Hello everyone and welcome back to C.A.R. Entertainment. I'm your host Dylan Hamilton today, guys, and we are going to be moving on with our A22 biology revision videos, guys. Last time we talked about ATP, and today we're going to be moving on and talking about aerobic respiration, which is going to be a very lengthy video, as you've seen by the time, and that's because we're going to go through our PowerPoint, and then I've got A3 sheets talking about the process of glycolysis, the link reaction, the Krebs cycle, and the electron transport change, which is the best way to learn this, actually. So basically, if you don't understand anything I'm talking about here, just wait to the end of the video and watch that 8-minute bit going through those sheets. Then it'll make sense. So I'm just going to go through this quite quickly, because it is better to learn it off the A3 sheets. So, an introduction. Well, the conversion of glucose into ATP is during respiration, and the cellular respiration of glucose is a complex integrated process which is divided into 4 stages. This is glycolysis, which is the splitting of glucose into 2 free carbon pyruvate molecules. The link reaction, which is the conversion of pyruvate into 2-carbon acetyl coenzyme A molecules. Krebs cycle, which is the feeding of acetyl-CoA into the cycle of oxidation and reduction reactions, or redox reactions. And then the electron transport change, which is the use of electrons to synthesize ATP. So glycolysis then, let's go through the theoretics of glycolysis. Well, it takes place in the cytoplasm, and glucose will enter by passive transport using carriers. The activation of glucose by phosphorylation makes glucose more reactive, and 2 phosphates are required to come from hydrolysis of 2 ATP molecules. Therefore, glucose phosphate will not fit the membrane and can't leave the cell. So following the first phosphorylation and fructose biphosphate, following the second phosphorylation, we have 6-carbon fructose biphosphate, which was split into 2 carbon molecules of triose phosphate. The triose phosphate is then oxidized through the loss of 2 hydrogens by dehydrogenation, which forms glycerate biphosphate. Hydrogen will then be collected by NAD, becoming reduced to form NADH, and the removal of hydrogen involves dehydrogenase enzymes and dehydrogenation. The conversion of each glycerate biphosphate into pyruvate molecule produces 2 ATP molecules, and each glucose will split to form 2 glycerate biphosphate, producing 2 ATP molecules for each glycerate biphosphate, and therefore there is a net gain of 4 ATP molecules. Or sorry, there's a gain of 4 ATP molecules, but there's a net gain of 2 ATP molecules because you've used 2 up in making the ADP at the start. So, the second phosphate lost from each glycerate biphosphate will form another ATP, and the production of ATP here is called substrate level phosphorylation, as ADP phosphorylates using the phosphate to form from compounds of the glycolysis pathway. Again, this will make a lot more sense when we talk about the sheets. So, the energy budget of glycolysis then. Well, the final products of glycolysis are pyruvate, ATP, and NADH, which is used to generate more ATP in the electron transport chains located in the mitochondria. NADH is unable to pass across the mitochondria, and therefore it is overcome this by molecular shuttles which carry electrons across the membrane. Glycolysis does not involve oxygen, and 2 molecules of NADH go to the electron transport chains. Pyruvate will go into the matrix of the mitochondria by active transport. So the link reaction then. Pyruvate will be transported into a matrix of the mitochondria and converted to acetyl coenzyme A, where pyruvate will be decarboxylated with the removal of 1 CO2 molecule. Dehydrogenation will take place with the removal of hydrogen and form an NADH, resulting in a 2-carbon acetate combining with coenzyme A to form a 2-carbon acetyl CoA. This will then enter the Krebs cycle, which takes place in the matrix of the mitochondria. So when this 2-carbon acetyl CoA combines with the 4-carbon acid oxaloacetate to produce a 6-carbon acid in citrate. The decarboxylation of the 6-carbon acid of citrate will result in the formation of the 5-carbon acid oxoglutarate, which is involving the loss of a CO2 molecule. The decarboxylation of then this 5-carbon acid oxoglutarate will result in the formation of this 4-carbon acid oxaloacetate once more, and then this process repeats for the second molecule of acetyl CoA. And of course, this decarboxylation results in the loss of a CO2 molecule. All these reactions will involve dehydrogenation, and at 3 points hydrogen is released forming NADH, and at 1 point hydrogen will be picked up by FAD, which is flavin adenine dinucleotide, to form FADH2. So 1 molecule of ATP is produced by the transfer phosphate group from the intermediate compound to ADP, and this is called substrate level phosphorylation. So in total, at this point, we have 10 NADHs in total after glycolysis, the link reaction, the Krebs cycle. We have 4 ATPs and a net gain after glycolysis, the link reaction, the Krebs cycle. And in the Krebs cycle, we have 6 NADHs produced and 2 FADH2s produced, and we have 2 ATPs produced, and this is because the 2 acetyl-CoA molecules will go around. So the oxidative phosphorylation, what is this? Well, it's a coupling of electron transport and ATP production in the presence of oxygen, where electrons will from NADH or NaH or FADH will pass through a series of carrier molecules called the electron transport chain. So electron transport chains then, well, it's based in the crusty of the mitochondria, and the ETC energy and hydrogen is converted into ATP. Coenzymes and many of these enzymes involved will attach to the inner mitochondrial membrane, which is when it's more deeply enfolded crusty, then it'll have a more extensive ultrastructure existing for ATP production. NAD and FAD will be arranged in a sequence of decreasing potential energy, with hydrogen moving along the chain, with carriers being successfully reduced and oxidized in a series of redox reactions. NAD and FAD will operate as hydrogen carriers. Hydrogen will move along the NAD to flavoprotein and coenzymes Q, and following coenzyme Q, the hydrogen will dissociate into electrons and protons. The electrons will pass along cytochromes in redox reaction, with the final hydrogen acceptor being oxygen, which will combine with these electrons and protons to form water, and the final stage of the ETC, it will be catalyzed by the enzyme cytochrome oxidase. A large protein complex will lie at progressively lower energy levels, with the energy becoming available as redox reactions take place. At certain points, there will be enough energy to produce ATP by oxidative phosphorylation, which you will see in our free sheets. So, oxidative phosphorylation and ATP production. Electrons will pass along the chain and lose energy, and as energy is used to pump protons through the inner mitochondrial membrane, a proton concentration gradient will be set up across the inner mitochondrial membrane, acting as a store of energy. It will be released if protons are allowed to pass back into the matrix, and protons re-enter through protein channels, where energy is used to synthesize ATP from ADP, which is catalyzed by ATP synthase. Mitchell's chemiosmotic theory is that electron transport and ATP synthesis are coupled by oxidative phosphorylation. ATP is produced, the total produced is 38, and that is a theoretical max as for each molecule of glucose. The value is actually likely to be closer to 30, because NAD is also used as a reducing agent for other metabolic compounds, and energy is used in transporting the reduced NAD from the cytoplasm into the mitochondria. Other respiratory subjects is that, and the equation for respiration is C6H12O6 plus C plus 6 CO2, giving 6 CO2 plus 6 H2O plus energy, which is your 38 ATP theoretical max. But your other substrates is that when glucose supplies are low, you'll have to use fat. So triglycerides will be hydrolyzed into glycerol and fatty acids, with glycerol entering the glycolysis stage and fatty acids entering the acetyl-CoA stage. Proteins can be hydrolyzed into amino acids, with the residue entering as acetyl-CoA or pyruvate, or one of the compounds in the Krebs cycle. Fructose can be broken down to glucose before entering glycolysis as well. Acetyl-CoA is called the hub of various respiratory pathways. I'm now going to transfer over to Dylan on the sheets, and I'll see you then.
Okay guys, so now we've went through the PowerPoint. I'm going to go through these two A3 sheets here. Not exactly the best quality, but whenever we zoom in it will be a wee bit better. So we'll be going through glycolysis, the link reaction, then the Krebs cycle, and then finally a wee bit about the electron transport chain. Sorry for my handwriting being horrific, but I will be talking you through it as we go. So start off with glycolysis. You start off with one molecule of glucose. So this is all from one molecule of glucose. Now, it is transformed into glucose phosphate, which are both 6-carbon molecules, and ATP is moved into ADP. So you're essentially removing that phosphate. Then this glucose phosphate is moved into fructose phosphate in a process called isomerization, which is whenever the atoms are rearranged as they have the same molecular formula but a different structural formula. Then phosphorylation takes place, and fructose biphosphate forms, which is an unstable molecule that splits into two. Now this allows ATP again to be formed into ADP plus your extra phosphate. Then you move on, and this splits then from fructose biphosphate into two molecules of triose phosphate, which are both 3-carbon molecules. So it goes from a 6-carbon molecule in fructose biphosphate to triose phosphate, which is 2 of them. So for one molecule of glucose, you eventually then get to 2 molecules of triose phosphate, where oxidation will occur through the dehydrogenase enzyme in the process of dehydrogenation, where NAD will then help this and take on the hydrogen, which forms NADH. And this means that triose phosphate then becomes glycerate biphosphate. So then glycerate biphosphate then carries out substrate level phosphorylation to become pyruvate, as we see 2 ADP becomes 2 ATP. So now we've produced ATP on each side. So from one molecule of glucose we now have 2 ATP produced. So we have 4 ATP in total at this point, with a net gain of 2. Okay, so then pyruvate becomes acetate in the link reaction, which is in the matrix of the mitochondria. So pyruvate then enters the link reaction, where by decarboxylation it will become acetate, where CO2 will be removed, and NAD will take on will become NADH. So then acetate will become acetyl-CoA, as the coenzyme A will be added, and then a 2-carbon molecule is formed. Sorry for the shaky hand as well, guys. It's very, very cold right now. So then acetyl-CoA moves into the Krebs cycle. Now, the Krebs cycle occurs twice for every single molecule of glucose. So for one molecule of glucose, we've now produced 2 molecules of acetyl-CoA, which then goes through the Krebs cycle. So we have acetyl-CoA coming in, and then the coenzyme A will leave, okay? And then you are left with your 2-carbon molecule combining with this 4-carbon molecule called oxaloacetate, which then gives citrate, which is a 6-carbon molecule. This 6-carbon molecule then is decarboxylized as CO2 is removed, and dehydrogenation occurs, meaning an NADH is formed. Then this decarboxylation forms oxoglutarate, and which is then also decarboxylated itself through the loss of CO2, and then there is a process of substrate level phosphorylation, as ATP is produced once in the Krebs cycle, with NAD being formed by dehydrogenation twice, and then a new hydrogen acceptor called FAD will be formed into FADH2 through dehydrogenation. And of this decarboxylation of oxoglutarate as a 5-carbon will then form right back to oxaloacetate as a 4-carbon. So at this point we have 6 NADHs and 2 FADH2s in the Krebs cycle that will move on to the electron transport chain, and 2 ATPs are made in the electron transport in the Krebs cycle, due to the fact that it occurs twice for every 1 molecule of glucose. So at this point then, in total, we have 10 ADHs that are going to go to the electron transport chains. 4 ATPs are the net gain. The 4 ATPs are produced by FADH2, and therefore there's going to be 30 ATPs produced by NADH, and in total we're going to end with 38 ATPs. This is after the electron transport chains, these 3 bottom ones. So let's go through the electron transport chains so you understand how these come about. So in the electron transport chains, you see up here NAD is NADH, and it will enter and it will come in contact with reduced flavoprotein, which can be reduced and oxidized to become flavoprotein. And in this redox reaction, the energy used means ADP can be converted to ATP, and this is called oxidative phosphorylation, whenever the energy in a redox reaction is used to create ATP. Now, as you notice, all these things called flavoprotein, coenzyme Q, reduced cytochromes, etc., they're all listed down in a diagonal, which is lowering levels of energy potential. So each time you go down, there's less energy. So the NAD will then move down from the flavoprotein into coenzyme Q. There's not enough energy for oxidative phosphorylation at this point, and therefore there is no ATP produced. FADH will come in at this point. It does not go into the flavoprotein, but instead just the reduced enzyme Q, which is a redox reaction. Now, at this point here, whenever it moves from the reduced coenzyme Q into the cytochromes, there is enough energy for oxidative phosphorylation, and therefore ATP is produced. Now, also at this point, hydrogen will dissociate into electrons and protons. So as we see over this dashed line and here over, that's where the protons go. But electrons will move on to the cytochromes, and they're accepted by cytochromes, where this redox reaction also forms enough oxidative phosphorylation. So as you see from here, we see ATP is produced for oxidative phosphorylation. So once they're then moved on then to reduce cytochrome oxidase, this is also a redox reaction. So at this point we have our 3 oxidative phosphorylations forming 3 ATPs for simply 1 molecule of glucose as 1 NADH. This is 1 NADH that comes in, and it's producing 3 ATPs through oxidative phosphorylation at reduced flavoprotein, cytochromes, and reduced cytochrome oxidase. Now, where does the protons come in? Well, protons are accepted by oxygen. This is where oxygen comes in aerobic respiration. So the protons and electrons are taken by oxygen to form water, and then water is released as a waste product. And that's essentially how the process comes. Now, because we have 10 NAD hitches coming into the electron transport chain, for every one you produce 3 ATP. So it's just 10 times 3, that's 30. That's where you get your 30 ATP, which is produced by NAD hitch. Now, because your FAD only comes in here, it only produces 2 ATPs. And because there was only 2 FAD hitches produced, that means it's 2 times 2, which is 4. So you have 4 ATPs, which are produced by FAD hitch 2. You add the two together, and you have your theoretically 38 ATPs, which are produced. This is going to be quite a long video, but I hope you have enjoyed it. I've been Dylan Hamilton from C.A.R. Entertainment, and I will see you next time.